humans. So now, as we said, the cell requires certain things in order to survive, to be alive, right? Those nutrients which we eat gets digested. That digested nutrients will be absorbed in the small intestine, and from there it will be passed on to the blood, right? Now from the blood, there are certain things which has to be supplied to other parts of the body, and they get diffused from the blood vessel to the cell where it is required. And the second is, it is also required to remove waste. So if you eat something, there is definitely the waste that is being generated in the body, and that waste in the form of CO2 has to be expelled out of the body. Now, where exactly this gaseous exchange takes place in the human body? Structure, that is a pair of lungs, and this lungs are supplied with more of this alveoli-like structures, and specifically if you enlarge one alveolus, you can see that whatever the nutrients and the waste that are present in the blood will be exchanged at this capillaries of alveoli. So what it does, the waste in the form of carbon dioxide is more in the blood, right? That will easily diffuse because it is in more concentration in the blood to the alveoli where it is less. Similarly, when we breathe in, the oxygen which is more in the air will diffuse from the alveoli. It will diffuse into the blood where it is less. So this exchange of gases occurs because of diffusion. We also see that once the food is digested in the small intestine, the food gets absorbed, especially in the villi of the small intestine, because of diffusion. Now we have seen in animals. Let's see what happens in plants. We know that it requires nutrients, and especially the root of the plants will absorb water. Since soil is having more water compared to the roots, water is going to diffuse through these roots into the plant, and from there it is transported to other parts of the plant. Water and minerals get absorbed because of diffusion. Okay, now where else you can see this process also helps in photosynthesis and respiration. In respiration, the exchange of gases takes place, and in photosynthesis, the raw materials, whatever is there in the form of CO2 and H2O, will diffuse because of this process. You must have seen that most of the times, if you forget to water the plant, what may happen? You see that the plant is almost in a drooping stage. You can see the leaves are folded or as if there is no life in that. Now what happens if you water the plant? So when you properly supply the water, you see the plant as fresh, right? So what is necessary? Watering of plant is necessary. Due to the process of osmosis, the water is taken up by the plants, and this water gives the plant a turgid structure or the firm structure. When it is wrinkled, the water is lost, and in turn the turgidity of the plant is lost. Do you find any other application of osmosis? Yes. We also see them in food preservation process. This process like pickling, drying. So this osmosis process where we add more of salt or sugar to preserve the food follows this process. Now let's see what happens if we are going to change the amount of solute in water where a plant or animal cell is placed. Do they find any difference? As we said, the movement of water molecules from a region of higher concentration to a region of its lower concentration, it depends upon the amount of solute that is dissolved in water. Let's see, there are three instances. So now this ability of the extracellular solution or the surrounding solution to make water move in and out of the cell by osmosis is affected. Now, what will happen if we are going to change the environment of a cell where it is placed by either adding salt or sugar solution? Let's see. Now, let us understand the terms here to understand what is the tonicity or the concentration here. Hyper means higher salt concentration or higher solute concentration in the external environment, okay, than in the cell where it is placed. Hypo means lower or less amount of solute dissolved in the external environment of a cell or outside the cell. The next is isotonic. That means the amount of solute that is dissolved is same inside of the cell and in the surrounding of the cell where it is being placed. Now, how does this tonicity or the concentration affect in the living system? Let's know that. So again, as we know, there are three different situations. One is hypotonic. What is the surrounding environment here? Hypo means the surrounding environment where the cell is being placed is having hypo means less amount of solute dissolved in the water outside than the cell. The water is more in this hypotonic solution. The outside of the cell has more water compared to inside. So what will happen as the process of osmosis? The water from the surrounding is going to enter the cell. Okay. Now next, hypertonic solution. Hyper means again the surrounding of the cell will have more of solute, in the sense less of water. So now where is the movement of water here? The cell is going to lose water to the surrounding. Is there any other central situation? Yes, it is. What it is? Isotonic solution. What happens in isotonic solution? The cell, so here the surrounding as well as the cell internal water content or the concentration is same both the sides. Therefore, this is known as isotonic solution. Let's see what happens in hypo. As the water enters the cell is going to swell up or it's going to increase in the size or bulging out. Now let's see what happens in hypertonic. So as more and more water will leave the cell or becomes flaccid. So now this process where the cell membrane along with the cytoplasm becomes small, shrinks with respect to the cell wall, we call that process as plasmolysis. And this happens because of loss of water from the cell due to hypertonic surrounding. So this process is called as plasmolysis. Do you think any change will occur in isotonic? No. There is no change in the cell structure as the molecules of water will move in both the direction in equal amount. For example, 4 molecules move out of the cell, then 4 molecules will enter into the cell. Therefore, there is no change in the size. The net movement equals same. Net movement of water will be same. Now let's see what happens if we place an animal cell, especially the human RBC, the red blood cells which are responsible to carry out oxygen. So these are the human RBC. You know they are concave in structure. Now if you are going to place an hypotonic solution, what happens? So the water enters into the cell to an extent that it keeps on increasing in size, just like a balloon. When you blow, it expands. Finally, it's going to burst to release its content out. Now, what happens in hypertonic solution? In hypertonic solution, the water will move out of the cell into the surrounding. Therefore, the cell size is going to shrink. So, what is ideal for a cell now? So, the ideal thing for this animal cell is it is safe to place an animal cell, human RBC, in the isotonic solution where the entry of water will be equal to exit of water. Okay. Now let's know why this is required. If we want to place a cell in isotonic solution, why it is required? It is required to maintain the internal balance, fluid balance of the cell and its content. Otherwise, the cell may die. So thank you. Thank you so much for listening to me.